Good afternoon, especially our instructor, Ma'am Julita Gatungay. My name is Heridity from BA Ed 1, and we are the Group 12. And so our topic for today is about the role of genes and heredity. The role of genes and heredity. A gene is a basic unit of heredity in a living organism. Genes come from our parents. We may inherit our physical traits and the likelihood of getting certain diseases and conditions from a parent. Genes contain the data needed to build and maintain cell and pass genetic information of offspring. What is genes? Genes, it is the basic unit of heredity, occupies a specific location on a chromosome. What is heredity? Heredity, it is the sum of all biology processes by which particular characteristics are transmitted from parents to their offspring. Heredity factors, known as genes, are thought to exist as the discrete portion. The term discrete refers to the concept that genes are always located at the same point of locus on a chromosomes. It believes that pair of homologous chromosomes contain linear, matching arrangement of genes exerting parallel control over the same traits. Pair of genes that exercise such parallel control over the same traits are known as alleles, which are the two kinds. It is dominant and recessive. What is the relationship between genes and heredity? Heredity and genetics are directly related. Genetic includes all the traits that you have that are encoded into your genes. These two copies of the genes contained in your chromosomes influence the way your cells work. The two alleles in a gene pair are inherited, one from each parent. Alleles interact with each in different ways. These are called inheritance patterns. How do genetics cause disease? Sometimes there is a mutation, a change in a gene or genes. The mutation changes the gene's instruction for making a protein. So the protein does not work properly or is missing entirely. Each gene carries instruction that determines your future. Here are some examples of common genetic disorder. First, Down syndrome. Down syndrome is a condition in which a person has an extra chromosome. Chromosomes are small packages or genes in the body. They determine how a baby bodies form and function as it grows during pregnancy and after birth. Typically, a baby is born with 46 chromosomes. Second, thalassemia. Thalassemia is an inherited blood disorder caused when the body doesn't make enough protein called hemoglobin, an important part of red blood cells. Third, cystic fibrosis. It causes severe damage to the lungs, digestive system, and other organs in the body. Cystic fibrosis affects the cells that produce mucus, sweat, and digestive juices. The body produces abnormally thick and sticky fluid called mucus. This mucus builds up in the breathing passages of lungs and the pancreas. Fourth, sickle cell disease. Sickle cell anemia is one of a group of disorders known as the sickle cell disease. Sickle cell anemia is an inherited red blood cells disorder in which there aren't enough healthy red blood cells to carry oxygen throughout your body. Fifth, hemophilia. Hemophilia is usually an inherited bleeding disorder in which the blood does not clot properly. This can lead to spontaneous bleeding as well as bleeding following injuries or surgery. Each of our gene contains unique protein. Protein has a separate and distinct function to perform in the cell. So, let's proceed to our next topic, and that is Loisa's report.
Thank you, Kyla Maybivar. So, first, let me just introduce to you myself. I am Loisa S. Villanueva. So, okay, before we start our discussion, so let me just remind you that we are talking about the role of genes in heredity. So now, as the discussion continues, we will now talk about the effects of genes in our environment. But before we start, I want to ask you, what is genes? According to my research, a gene is the basic physical and functional unit of heredity. O sa madaling salita, ang genes, ito ay minanana or minana. Ngunit naniniwala ka ba ng kanilang ekspresyon ay maaaring magbago? Nang dahil lamang sa kanilang pakikipag-ugnayan sa kapaligiran o pagkakaroon ng ugnayan sa kapaligiran? Okay, before I give you some example of these effects, let me just share you the main difference between these two terms lines in the fact that heredity have the potential of being carried from one generation to another, whereas a genetic can either be a heredity or not, but there will always be a mutation change in the genome. Okay, now let's move on to my preferred two examples. Okay, the first example is from the Bible of Genesis. I just abbreviated it, but this is what the content means. Ito ay patungkol kay Jacob at sa kanyang tiyo Laban. Si tiyo Laban ay may maraming alagang tupa. Ngayon, ang kanyang mga alagang tupa ay may mga buntis. Nagkaroon sila ng kasunduan na kapag ang mga buntis na tupa ay magkaanak na ang kulay ay may batik-batik or black spot ay magiging kay Jacob. Ngayon, ang ginawa ni Jacob ay naghanap siya ng iba't ibang uri ng punong kahoy at ito ay kanyang pinalatan na kung saan ito ay nagmukhang may batik-batik. Inilagay niya ito sa painuman upang makita ng mga hayop tuwing iinom. Dahil rito, ito ang napaglihian ng mga nagbubuntis na tupa. Dahil roon naapektuhan ng genes. Ang naging resulta nung pinanganak na ang mga tupa ay halos lahat ay may batik. So our next example is about the effect of light or chlorophyll production. Although most plants have the genetic ability to produce chlorophyll, they will also do this only in the presence of light. Without light, these plants produce only a light yellow pigment and therefore appear pale and sickly until they are exposed to the sun. Well, after a few days of exposure to sunlight, the chlorophyll production mechanism is enabled and green color return. Hey, and now we're going to talk about gene mutation. Okay, genetic mutation. Mutation is the result of copying mistakes made during cell division through the exposure to radiation. Chemicals called mutagens are infection by viruses. So, these mutations alters the genome which provides all the information the organism requires to function affects, each resulting in changes in the body, such as external or internal abnormalities. Mutational effects can be beneficial, harmful, or natural, depending on their content or location. So, most non-neutral mutations are deleterious. Also, mutations na accumulate over time is the result of evolution. A nucleotide consisting of a sugar molecule attached to a phosphate group and a nitrogen-containing base. The bases used in DNA are adenine, cytosine, guanine, and thymine. A nucleotide is the basic building block of nucleic acid. Nucleotides are in a particular essential for application of DNA and transcription of RNA. in rapidly dividing stages. Ribonucleic acid or RNA is a molecule similar to DNA. RNA is a single-stranded. An RNA strand has a backbone made of alternating sugar and phosphate groups. All known cellular life and some viruses contains DNA that contains the genetic instructions for the development and function of living things.
Simple terms. Example, during exam teacher equals nucleic acid test paper equals genetic information RNA equals student test results equals proteins mutations equals mistakes test score equals function or a living being.
Hi, my name is Janica de Andres. I'm from BE 1. Let's now proceed to my topic, which is DNA and genetic code. First, we will discuss what is DNA. DNA stands for deoxyribonucleic acid. It is the molecule that contains the genetic code of organism. This includes animals, plants, protists, archaea, and bacteria. DNA is in each cell and the organism tells cells what proteins to make. They do not code for protein sequences. Deoxyribonucleic acid is more commonly known as DNA. It is a complex molecule that contains all of the information necessary to build and maintain an organism. All living things have DNA within their cells. In other words, whenever organisms reproduce, a portion of their DNA is passed along to their offspring. What is DNA made of? DNA is a linear molecule composed of 4 types of smaller chemical molecules called nucleotide bases. First is adenine, cytosine, guanine, and thymine. The order of these bases is called the DNA sequence. The genome is mostly the same in all people, but there are variations across the genome. This genetic variation accounts for about 0.001% of each person's DNA, and it contributes to differences in appearance and help people who are closely related have more similar DNA. Cell nucleus is nearly that every cell in person's body have the same DNA. Most DNA is located in the cell nucleus, where it is called nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA. The diploid human genome is thus composed of 46 DNA molecules of 24 distinct types. Because human chromosomes exist in pairs that are almost identical. Only 3 billion nucleotide pairs, or the haplogenome, need to be sequenced to gain complete information concerning a representative human genome. How does DNA control our life? DNA contains the information to make protein, which carry out all the functions and characteristics, which are essentially determined by proteins. So, DNA contains the instruction for making a protein. What is DNA and its function? DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce a proteins, which are the complex molecules that do most of the work in our bodies. And also, DNA code contains instructions needed to make the proteins and molecules essential for growth, development, and health. The sequence of the bases E, C, G, and T In DNA, determines a unique genetic code and provides the instructions for producing molecules in the body. The cells reads the DNA code in group of 3 bases. Each triplet of bases called codon during protein synthesis. 20 different amino acids, which are the building blocks of proteins. Different proteins are made up of different combinations of amino acids. This gives them their own unique 3D structure and function in the body. Only 61 of the 64 codons are used to specify which of 20 amino acids is next to be added. There are 3 codons that don't code for amino acids. These codons mark the end of the protein chain.
Here on the screen, I have a big string of RNA. This is RNA. And RNA is the code that creates proteins inside organisms. A big string of RNA just might look like this, a big gobbledygook of a whole bunch of different letters. But RNA is actually organized into groups of 3, like this. And we call each group of 3 a codon. And actually what these are groups of is each individual one of these letters is called a nucleotide. Nucleotide. And so a group of 3 nucleotides is called a codon. And what each of these codons do is each of these codons codes for an amino acid. So AUG here codes for an amino acid. Amino acid 1, I'll just put a 1 here. With each of these codons coding for a different amino acid, what happens is all the codons put together code for a protein. A protein. Oh, protein. All right, so all of the amino acids put together create a protein. And here, this is amino acid number 7. I'll put amino acid here so that you can remember that each codon codes for an amino acid. One other thing that's really important to remember about RNA here is that because these 7 codons put together create a protein, it's really important for RNA to know when the beginning of a protein starts and when the end of the protein is reached. And so the beginning here and the end of these proteins need to be signaled. And how codons and RNA do this is they have certain universal amino acids, certain codons here, certain codons that code for a protein to start and for a protein to stop. And so here at the end here, this is a stop codon that says, hey, this is the last codon. You can end here and finish off the protein. So UAG, that's a stop codon. And at the beginning of our list here, we have a start codon. AUG says, hey, this is the beginning of the code for a protein. I am a start codon. So AUG is a start codon, and UAG is a stop codon.
The codon wheel above can be used to translate DNA codons into amino acids. Find the first letter of your sequence in the inner circle and work outward to see the corresponding amino acid, for example, ETG, methionine. Let's now come to genetic code. Genetic code is pertaining to the set of rules defining how the 4-letter code of DNA is translated into the 20-letter code of amino acids, which are the building blocks of proteins. There are 64 possible permutations or combinations of three-letter nucleotide sequences that can be made from the 4 nucleotides. So that's all for today. Hope you enjoy and gain some knowledge about our topic. And again, thank you for your time and appreciation for our presentation.